everyone and welcome back to ByHollyG. Welcome to today's video, which as you can tell from the title is basically all about the COVID-19 vaccines. And the reason why I'm filming this video around this time, or on this day in fact, is because I literally had my vaccine a few hours ago this morning. I actually filmed myself having the jab for my vlog on my main channel, but yeah, I had my vaccine today and I just thought there was no better time to talk about the biology behind vaccines, and specifically the COVID-19 vaccines, because I feel like if you do get the vaccine yourself, you know, you might want to know a bit more about the biology behind it. I will also try to limit the amount of technical knowledge in this video so that anyone can understand and follow what I'm talking about. But yeah, I hope you enjoyed this video anyway. Definitely give it a thumbs up if you do, comment down below if you have any questions or video ideas, and subscribe obviously if you want to stick around and join the ByHollyG community for lots more biology content in the future. And I should actually say as well, these two dots on my face are not a side effect of the vaccine. I decided to attack my face clearly, and I'm not covering them up because I thought you might want to stare at them the whole of this video. But anyway, random side note, we're gonna move on. So basically in this video, as I said, I'm gonna talk about vaccines and the COVID-19 vaccines, but I'm specifically gonna talk about the Pfizer and the Moderna vaccines because they use a very new vaccine technology based on mRNA. Obviously, these vaccines have been approved very quickly. It normally takes a lot longer. It can take up to like 10 years to approve a new drug or a vaccine, but I just think it's a very clear demonstration of, you know, how rapidly science can advance. And I just think it's a huge success story. Like, it's such an incredible achievement in the field of biology and science, you know, to have approved vaccines, multiple vaccines now, to be used against the virus. So the first question we're going to answer is basically, what is a vaccine? And I'm going to be talking specifically about prophylactic vaccines. Now, what prophylactic means is that it is something to be used as a preventative measure. So a prophylactic vaccine is something that is given to a healthy individual in order to prevent them, hopefully, from getting the actual disease. And that is in contrast to therapeutic vaccines. So for example, like anti-cancer vaccines, which are not usually given to prevent someone from getting cancer, but they're usually given to treat someone who is already ill and who already has cancer. So most of the time when people say vaccines, they are talking about prophylactic vaccines, those that are preventative. But yeah, I just wanted to make that apparent just in case you were wondering. So basically, starting off with a definition then, a vaccine is an agent that basically stimulates the immune system to recognize, remove, and remember that agent that was injected. And that memory component about vaccines is most important. You know, as a result of giving someone a vaccine, we are generating immunological memory in that individual. So this agent that is injected can take on different forms. So sometimes we use a whole pathogen. Now, a pathogen is basically just a microorganism that causes disease. So for example, like a bacterium or a virus that causes a disease. But most importantly, that agent has to be either dead or it has to be weakened, or what we say is attenuated, so that it doesn't cause the full-blown disease, obviously. The whole pathogen could also be a related pathogen, so it's not the pathogen itself, but it's a related pathogen that is harmless. And if we don't decide to use the whole pathogen, then we can use a subunit of that pathogen. And we also have vaccines that are based on bacterial toxoids, which are basically where we inject a particular toxin that is released by a pathogenic bacteria. And for the rest of this video, I'm going to refer to that injected agent as the antigen, because an antigen is just a generalized term for anything that can stimulate an immune response. What happens when we give someone a vaccine is we stimulate what's called a primary immune response. Now, a primary immune response is the immune response generated when you first encounter a particular pathogen. The immune system as a whole is very complex. The immune response is also very complex, but the immune response basically has two main components. We have this first and very rapid innate immune response, which is what we call the non-specific response because it's quite generalized. And the second type of immune response is arguably more sophisticated because it's specific to the invading pathogen. So for example, COVID-19, it can specifically recognize it as COVID-19, and it takes longer to generate. But this specific adaptive immune response is what we call it. It leads to the production of two very important immune cells called our B cells and our T cells. Now, the former, so the B cells, they are responsible for releasing what we call antibodies. And for a whole video about antibodies, I made one recently. You can check it out. I will leave a link either up here or here. And it also leads to the production of T cells. And in a viral infection, both antibodies and T cells, specifically the cytotoxic or killer T cells, are very, very important in getting rid of the virus. So we need both of those cell types to be generated as part of this immune response. What happened? Did I just break the desk? Something that's really important to stress though is that a vaccine is designed to, yes, lead to this primary immune response, and there might be some side effects as a result of that immune response, but the vaccine is not designed for you to get and contract the actual disease. That would be a disaster if a vaccine leads to the actual disease, right? And going back to what I said at the start, what we inject in a vaccine is something that is ultimately harmless. It's either dead or it's weakened or it's just a subunit of the pathogen. It's not the actual pathogen. So when you are given a vaccine, you're not going to get the disease. You are just injected with an antigen that is sufficiently immunogenic to generate an immune response and that memory component, but it's not going to cause the disease, most importantly, is what I'm trying to say. And then pulling this all together, I'm basically going to show you a graph to show you the primary and the secondary immune responses, because a vaccine basically takes advantage of this primary immune response, because the secondary immune response, as you can see on this graph, is a lot stronger and it's a lot faster. So the primary immune response, it's a strong response, it's going to get rid of that antigen, but it's not as strong as the secondary immune response, and it also takes a lot longer to be mounted, whereas the secondary immune response is faster and stronger. Those are your two key words to be associated with that secondary immune response. So when you've had the vaccine, you've generated a primary immune response. If you are then infected with the actual pathogen, the actual thing that's going to cause the disease, your body is going to immediately generate that secondary immune response that is faster and stronger, and so you won't now contract the full-blown disease. If you do get symptoms, you'll get milder symptoms, or you won't see any symptoms at all. So in a nutshell, you're basically protected, and that's because the primary response led to that memory response, and because of that memory response, your body remembers that original antigen. So if it sees it again, it will immediately generate that secondary immune response, which is generated by the memory immune cells. I don't want to give you too much information so that you get confused, but you can imagine a situation where in the primary immune response, when you've never seen a pathogen before in your whole entire life, say for example COVID-19, you've never been infected with COVID, for example, in your body there will be very, very few immune cells that can recognize COVID-19. So in the primary immune response, it obviously takes a long time because those specific immune cells have to see COVID, and it might take quite a long time for those cells and that pathogen to interact. However, once you've generated that primary immune response and you've generated memory cells, those memory cells circulate in much larger numbers now. So you have a much larger team of, I don't know, immune cells or policemen patrolling your body, so that when you get the actual disease and you mount that secondary response, it's a lot faster because you have a larger population of those memory cells. So it doesn't take as long for that interaction to occur between the pathogen and those memory cells. So it's not only faster, but it then is also stronger as well. So before we go on to talk about the actual COVID mRNA vaccines, I also just wanted to give you a little bit of background about COVID itself, just in case you didn't know much about it. Basically, COVID-19 as a disease, the pathogenic agent behind COVID-19, or the pathogen that causes the disease, is called SARS-CoV-2, and that stands for Severe Acute Respiratory Syndrome Coronavirus 2. So as we already know from the pandemic, it is highly transmissible. It causes disease symptoms that can be very variable, and obviously it emerged in China in late 2019, and you know, since then it's spread around the world. I think in March 2020 the World Health Organization officially declared it a pandemic. I'm not going to dedicate this video entirely to viruses, obviously it's about vaccines, but a virus is non-living, and a virus at its simplest is basically genetic material, either DNA or RNA, enclosed in a protein coat, which is what we call the capsid. Coronavirus is a bit more complex than that. Its genome is RNA, it's not DNA. It is enclosed in a capsid, obviously, and it also has an envelope around it outside, and it has a few other things going on here and there, but I'm not going to talk too much about that. A viral genome, as I said, it can be DNA or RNA, but for coronavirus it is RNA. It encodes a variety of viral proteins that ultimately build the virus, And for coronavirus, there are lots of different viral proteins, but the one I want you to focus on is what we call the spike protein, or it's denoted as just the letter S, because that is the protein that is used in the majority of COVID-19 vaccines. And the function of that spike protein is basically to allow the virus to enter host cells, because viruses, in order to replicate, they need to replicate inside a host cell. They cannot do anything if they're outside a cell. They have to be inside a cell so that they can make loads of copies of themselves. And in order to get into that cell, the coronavirus or COVID-19 uses its spike protein. The analogy for you guys today is basically that spike protein being a key, which basically unlocks a cell to let COVID-19 into the cell so it can replicate itself, make loads of copies of itself, and then spread, and so on and so forth. So the third and the main part of this video is all about the 2 mRNA COVID-19 vaccines, so those being Pfizer and Moderna. I am specifically going to talk about Pfizer. I actually got the Pfizer one, so we're talking about Pfizer, but Moderna and Pfizer, because they are both based on mRNA, they work in very similar ways. And the really cool thing about mRNA vaccines is that it is very new technology. There hadn't been any mRNA vaccines approved before the ones against COVID-19, so it is very new technology. And the crazy thing is that I remember this being discussed when I was at Cambridge and doing my undergrad, and you know, everyone was really excited about mRNA vaccines because they're a new type of vaccines. Most vaccines will use the protein itself and not the mRNA, as I'm going to go on to explain. If you haven't studied biology, you're probably wondering what even is mRNA. Now, mRNA, it stands for messenger RNA. So if you want a bit more background, you can watch the video that I made about cells and what's inside a cell. But for now, what you can think about is inside a cell, we have the nucleus, which is where we find the DNA. And that DNA is basically the instructions for a cell. It tells a cell which proteins to make, and with those proteins, the cell can perform a particular function. And in biology, the process of making those proteins, so from going from DNA to protein, it's not a direct process. It takes 2 steps. Now, the first step is basically what we call transcription, and the second step is what we call translation. In transcription, first of all, in the nucleus, the DNA is converted into or transcribed into mRNA, and that mRNA acts as a messenger. It basically moves out of the nucleus and into the cytoplasm, where it can be translated into a protein on a structure that we call the ribosome. As I said, for more information about cells and what's inside them, you can watch that other video. I still don't know which side it's going to be on. I've done YouTube for such a long time, I can't actually believe I don't know where it is right now. So this 2-step process, going from DNA to mRNA to then protein, that is what we call the central dogma of molecular biology. It's such a mouthful and it sounds really fancy, but it was this original dogma, or this very key part of cellular biology. But for now, I just want you to remember that protein synthesis is a 2-step process, and mRNA acts as that intermediate messenger molecule. It basically carries the instructions for making that protein, and it's taken those instructions from DNA. So in this Pfizer vaccine, the agent that is injected is basically mRNA that encodes the spike protein, or a particular portion of the spike protein. If you want to know the details, the mRNA in the COVID-19 Pfizer vaccine carries the instructions for making the receptor binding domain of the spike protein. So as I said, that spike protein on COVID-19 allows it into a host cell, and it has a particular portion that specifically binds to a receptor on a cell, so we call it the receptor binding domain. And it's that small portion of the spike protein that is coded for in the mRNA that is used in the Pfizer vaccine. And this mRNA is basically packaged inside what we call a lipid nanoparticle, and that's basically a tiny particle, hence nano, and it's made of lipids, which are kind of like fatty structures, you can think of it. So this tiny lipid nanoparticle encloses the mRNA, and what will happen is this lipid nanoparticle that is carrying the mRNA is basically going to fuse with a particular type of cell, and once inside the cell, once it's inside the cytoplasm where we have the ribosomes, and as I said, the ribosomes are the structures on which translation occurs, so mRNA becomes protein. So now we've turned that mRNA into the actual spike protein, or the actual portion of the spike protein that the mRNA codes for. And in short, that spike protein is going to be basically presented to the immune system. The immune system will see it as foreign, and so it's going to start mounting an immune response against it. It's going to remove that agent, but as I said at the start, most importantly, it's going to remember that agent. We're going to get memory cells that are going to remain circulating in your body in much larger numbers. So if you do actually get COVID-19, then you are going to be stimulating the secondary immune response that is a lot faster and stronger, so you won't get the actual disease. So as I said before, mRNA vaccines are very new. They're really cool technology, and they do have quite a few advantages. So the first thing about mRNA vaccines is that we can design them in such a way so that we know exactly what that mRNA is coding for. What instructions is that mRNA going to make? We can specifically design that in an mRNA vaccine. The second advantage of mRNA vaccines is that they can be made outside cells. They can be made in a process that's called in vitro transcription. As I said before, transcription is where we go from turning DNA into mRNA, messenger RNA, and this process is in vitro because it happens in the lab outside cells. And so because we don't have to rely on cells to make the mRNA for us, it can be done a lot faster, and we can make mRNA in very large quantities. So it's very good for the production side of vaccines, and it also lowers the risk of contamination because we're not using cells. Obviously, as I said, any successful vaccine has to be harmless. It cannot cause the actual disease, and mRNA is very good for this as well, because as I said, we can very clearly define what the mRNA codes for. And so in this case, the mRNA only codes for a small portion of the pathogen. It's that receptor binding domain of the spike protein of COVID-19 in this case. And because that's only a small portion of the pathogen, it's not the whole pathogen, we're obviously not going to get COVID-19, the full-blown disease. mRNA is also low risk because when it enters a cell so that it can be translated, it doesn't go into the nucleus where the DNA is. So there's no risk of that mRNA integrating into the DNA and causing havoc with the DNA, like causing mutations, what we call insertional mutagenesis, where it inserts into DNA and causes damage and disruption. There is no risk of that because it stays in the cytoplasm, which is where the ribosomes are, so it can be translated. In the body, mRNA is also going to break down quite easily. So, you know, it's going to generate that immune response, and it's eventually going to be removed from the body entirely, so it's not going to stay there. Some of the potential limitations of mRNA vaccines are firstly that mRNA sometimes can be very lowly immunogenic, and what I mean by that is that sometimes the mRNA is not kind of potent enough to trigger a strong enough immune response to generate immunological memory. Another potential disadvantage, sometimes the mRNA itself might be recognized by the immune system and seen as foreign. The way in which we solve this issue is we make the mRNA look like our natural mRNA, so it doesn't look like a foreign agent, and if we disguise it in that way, then our immune system is not going to flag it as something that's foreign and that's something we should mount an immune response against. The final thing I'll say about mRNA vaccines is that sometimes it's difficult to deliver the mRNA. However, as I said, for the COVID-19 vaccines, the mRNA is packaged inside a lipid nanoparticle, and that's because mRNA itself breaks down quite easily, which is good because we obviously don't want the mRNA to remain in your body for a long time, but you know, it needs to be there for long enough so that it is recognized and stimulates an immune response. So yeah, those are some pros and cons of mRNA vaccines. The last thing we're going to talk a bit about is herd immunity. So herd immunity is basically where enough of the population are vaccinated or immunized against a particular antigen, and so that means there are very, very few vulnerable individuals that can act as hosts of the pathogen. And so because there are so few hosts that the pathogen can spread in and cause disease in, it means that it will eventually just be eliminated. You know, it has a really hard time trying to spread because most people are protected. As I said before, it has to be the majority of the population vaccinated. Now, the precise figure varies. Normally it's around 95% of the population needs to be either vaccinated or immunized for herd immunity to be effective, but that figure does depend on the pathogen itself. For COVID-19, I've seen very varied figures, like anything from like 70 to 90%, so it does vary. But the bottom line is that a lot of people need to be protected in order for herd immunity to be successful. And a very good example of, you know, herd immunity at its best comes from smallpox. So smallpox was a disease. It's no longer a disease because it was eradicated, but smallpox is caused by a virus. And basically, when it emerged and when it was spreading, there was this kind of global effort to generate vaccines and to get enough of the population vaccinated and protected. Because we achieved herd immunity, you know, as I said before, there were very few hosts for the virus to spread in, and so it was just eliminated, it was eradicated. The thing with COVID that makes everything a lot harder is that this virus changes and it mutates really quite rapidly. There are new variants arising all the time, so this makes it a lot more complicated to achieve herd immunity and to eradicate viruses and stuff like this. But you know, we are moving in the right direction, and in summary of this video, obviously I hope you've learned about vaccines, but you know, there are new vaccines being developed all the time. We're also looking into developing drugs to cure the disease if people get COVID. But yeah, vaccines are incredibly powerful. There is very clever biology behind them. So yeah, that is everything for this video. I really hope you enjoyed it. Definitely give it a thumbs up if you did. Comment down below, subscribe, and also hit the bell so you know when I upload. Obviously I can't upload every week at this moment just because I am busy with my masters, but in the future there will be more frequent uploads and more biology content your way, so yeah, stay tuned for that. And yeah, I will see you very soon in another video on ByHollyG. Bye!